Hello, everyone. My name is Romal Verma. I'm an undergrad at the University of Michigan and work with Dr. Chen and Buras to study the impact of metabolic changes on skeletal muscle biology. Our group recently showed that obesity causes anatomic remodeling and contractile dysfunction of the diaphragm in obese mice. Today, I'd like to make the case that a circulating extracellular matrix protein called Thrombospondin-1 contributes to these pathologic changes and might therefore be an important regulator of breathing impairment in obesity. Respiratory dysfunction is a major problem for obese individuals. 40 to 80% of these patients are affected by overlapping conditions that include dyspnea under exertion, obstructive sleep apnea, and obesity hyperventilation syndrome, or OHS. Obesity-associated respiratory dysfunction is often attributed to mass load, compression of the chest cavity and airways by excess adipose tissue. Reduced central nervous system response to hypercapnia has also been implicated in OHS. Respiratory muscle dysfunction occurs clinically in obese and diabetic individuals. However, the impact of the obesity on respiratory muscles, in particular the diaphragm, is now well defined. And in the lab, we use mouse models to study how obesity affects diaphragm function and structure. In vivo, you can use ultrasound to track diaphragm motion. This can be integrated over time to create an M-mode plot with diaphragm displacement on the y-axis and time on the x-axis. On these plots, you can measure diaphragm excursion amplitude, inspirative velocity, and expirative velocity. To determine the functional changes in the diaphragm itself, you can isolate muscle strips, electrically stimulate them, and study their contraction in ex vivo. This allows measurement of specific force, which is the isometric contractile force divided by the muscle cross-sectional area. We subjected normal mice to a 6-month high-fat diet time course and compared them to chow diet-fed controls. As expected, body weight progressively increased in the high-fat group. Amplitude, inspirative velocity, and expirative velocity were significantly lower in the high-fat mice after 6 months of feeding. Critically, at the 6-month time point, specific force was significantly lower in the high-fat versus chow-fed mice, and the 15% decline seen here is about half of what you'd see in the diaphragm of a mouse with muscular dystrophy. Checking out the tissue histology, we did not see overt atrophy, but did observe significant adipocyte accumulation with long-term high-fat feeding. In parallel, polymerized collagen deposition, seen here as orange red densities on serous red staining, was higher as well. Quantitatively, both adipocyte number and polymerized collagen content progressively increased with duration of obesity and were significantly higher in high-fat versus chow mice at 6 months. In strips subjected to isometric testing, both parameters negatively correlated with this specific force. We wanted to determine the source of this adiposity and fibrosis in the obese diaphragm and focused on fibroadipogenic progenitors, a skeletal muscle mesenchymal stem cell population that can differentiate the adipocytes and fibroblasts. In normal muscles, FAPs are important for satellite cell differentiation, myofiber maintenance, and repair. In pathologic conditions such as muscular dystrophy and injury models, they can contribute to pathologic adiposity, fibrosis, and muscle degeneration. FAPs are uniformly positive for PDGFR-alpha and can also be identified by this classic flow cytometry marker profile. Using these markers, we can isolate FAPs by flow cytometry and grow them in culture, their identity confirmed by PDGFR-alpha staining. In vivo, lineage tracing showed all adipocytes and many collagen-depositing cells arose from FAPs. High-fat feeding caused FAPs to increase in overall number and proliferative capacity. FAPs isolated from high-fat mice also showed increased expression of collagen species. These changes were not observed in non-FAP stromal cells, suggesting that FAPs are a key source of collagen deposition in the obese diaphragm. In the culture dish, FAPs isolated from high-fat diaphragms showed increased proliferation and collagen deposition. They did not show increased adipogenic potential, suggesting that the higher adipocyte numbers seen in the obese diaphragm is a result of a larger FAP pool. So we tried to determine the mechanism underlying diaphragm dysfunction, remodeling, and FAP changes, and focused on a matricellular protein called Thrombospondin-1. It is secreted by macrophages and adipocytes, circulates in the blood, and functions in wound healing. It is a potent stromal cell mitogen and activates TGF-beta and PTGF-A, which are critical FAP growth factors. It is implicated in inflammatory myopathy pathogenesis, and its levels increase with obesity. All these properties made it a good candidate for the molecular regulation of obesity-associated diaphragm remodeling. In our diet-induced obesity model, THBS1 increased in the plasma. Treating isolated FAPs with THBS1 caused dose-dependent increase in proliferation and collagen deposition. You can see here that the high-fat group also had increased collagen deposition at baseline. To determine its in vivo impact on diaphragm biology, we subjected wild-type and THBS1 knockout mice, 6 months of high-fat feeding. Knockout mice weigh slightly less than wild-type. However, a known feature of this knockout is a slightly smaller size. And as you can see, body length was also lower. Therefore, body composition was equivalent between wild type and knockout mice in both chow and high-fat diet. Ultrasound measurements showed a nice staircase decline in amplitude, inspirator velocity, and expirator velocity in wild type mice during the diet time course. And in the knockout mice, all parameters stayed essentially the same over 6 months of diet-induced obesity. Critically, specific force was higher in the knockout versus wild type diaphragm at the 6-month time point, demonstrating protection from obesity-induced diaphragm dysfunction. We histologically analyzed diaphragm samples from wild type and knockout mice at 6 months and saw that knockout animals had reduced adiposity, as indicated by paralipin staining. Quantitatively, adipocyte size and number, as well as the total cross-sectional area occupied by adipocytes, were all lower in the knockout. Adipocyte occupied area also negatively correlated with specific force. In addition, knockout diaphragms had reduced collagen deposition and lower expression of genes related to extracellular matrix deposition. More broadly, RNA sequencing and gene set enrichment analysis showed ECM-related pathways to be among the most strongly enriched in diaphragms of wild-type mice. In addition, analysis of a curated set of established FAP markers showed these to be predominantly higher in wild-type versus knockout. Consistent with this, preliminary flow cytometry also showed FAP number to trend higher in diaphragms of wild-type mice. In summary, physiologically, diet-induced obesity impaired diaphragmatic motion and reduced asymmetric force, and both of these were protected from the THBS1 knockout. Anatomically, diet-induced obesity increased diaphragmatic adiposity and fibrosis, both of which were once again reduced in the THBS1 knockout. And at the cellular level, diet-induced obesity increased FAP proliferation, collagen gene expression and deposition, and THBS1 itself was responsible for FAP proliferation and collagen deposition. Therefore, in the THBS1 knockout, we saw reduced FAP-related gene expression and FAP number. Overall, we believe THBS1 is a novel player in diaphragm remodeling and dysfunction in obesity, and better ascertaining its mechanistic role may enable novel translational approaches to managing respiratory dysfunction in obesity. Finally, I'd like to thank my mentors and collaborators for this study, and would love to answer any questions you may have, and you can contact me at the email provided here. Thank you, and have a nice day.